Good morning, everyone. My name is Alan Rodrigo, and it's an honor and a delight to introduce this morning's keynote speaker. It's also somewhat daunting because Sir Peter Gluckman has a long list of awards and accolades, and it's hard to keep them straight as I'm thinking about the introduction. Peter is a fellow New Zealander, so that in itself is a great pleasure, as I said, to introduce him. He is the Distinguished Professor of Pediatric and Perinatal Biology at the University of Auckland. That was my stomping ground until about 5 years ago. At various points, he's been the head of the Department of Pediatrics, the Dean of the Faculty of Medical and Health Sciences at the University of Auckland, Director of the Liggins Institute, and Director of the National Research Center for Growth and Development. Peter holds professorships also at the University of Southampton and at the National University of Singapore. In Singapore, he's also the Program Director for Growth, Development, and Metabolism at the Singapore Institute for Clinical Sciences. Peter has done a great deal of work internationally, working on his policy, working on evolution and development and how they relate to health. In 2014, for instance, Peter co-chaired the WHO's Commission on Ending Childhood Obesity. As I said, Peter has received several international awards, recognition. He is, for instance, a fellow of the Royal Society, the British Royal Society. He's also a fellow of the Royal Society of New Zealand. He's the only New Zealand member of the Institute of Medicine in the US. And in 2009, Peter was knighted in New Zealand for his services to medicine and science. Peter has written over 500 papers and several books focusing on a variety of topics germane to his research, including books on evolutionary mismatch and the fetal origins hypothesis. His research has focused not only on the proximate gestational and early life drivers of health outcomes, but also on the evolutionary explanations for why these drivers exist in the first place. Since 2009, Peter has been the Chief Science Advisor to the Prime Minister for New Zealand. And as New Zealand's Chief Science Advisor to the Prime Minister, Peter has a unique perspective on the interplay between science and policy. He has written several papers on the complexities of this interaction and on the sometimes conflicting explanations of the expectations of the lay public, the scientists, and politicians. That he remains now, after close to 6 years in the job, still highly regarded by the public, by scientists, and by politicians is, some would say, miraculous. And so today, I think, Peter is going to tell us about this interplay in his presentation. Peter.
Well, I'm actually going to disappoint you, Alan. I'm going to talk largely about evolutionary biology. A few months ago in Nature, this paper appeared. Well, it wasn't actually a paper. It is a discourse between two groups of prominent evolutionary biologists, people who think that the classic model since the modern synthesis is an adequate explanation of evolutionary phenomena, and people such as Leyland and others who think that there are fundamental issues in the current framing of the evolutionary synthesis and believe that a considerable rethink is needed. Now, fundamentally, when you look at this discourse in Nature, which is well worth reading, it really is an important discourse, what the two groups are really debating is the place of development within the evolutionary synthesis. I'm not going to dwell on that at length, but I'm just going to highlight some of the points as I move through this talk, because I think all of you should read that discourse. What are the issues that those who thought the evolutionary synthesis needed some rethinking pointing out? They were raising issues of the significance of non-genomic inheritance systems, They're raising issues how they might interplay with the genetic inheritance. The issues of what Mary Jane West Eberhardt first called phenotypic-driven evolution. But particularly they were talking about the role of adaptive developmental plasticity in terms of evolutionary processes. And of course that brings one into what we will talk about a bit in this talk, the interplay between genetic and epigenetic biology. They also raised a couple of issues, and I've added to that list a little bit, namely the fact that there are a number of biases in play in evolutionary processes, as we all understand. But one of them that tends not to be forgotten, often to be forgotten about, is the biases that adaptive developmental plasticity can put on systems. But I would also raise, and we'll come back to it perhaps if we have time, the whole issue of mutational biases in the genome, because of course methylated and particularly hydroxymethylated CPGs, at least at some points in the chromatin, not across the whole of the chromatin, are particularly mutable with long-term consequences. And then there's the whole issue of genetic assimilation, which I won't focus on today, but raises the interesting issue, does it really exist or does it not exist? And if it exists, how is it that what starts off as a phenotypic variation gets fixed in the genome? Is it by random mutation or is it by some other process? Just to remind you that there are a large number of non-genomic inheritance systems, and what's happened in the last 2 years is the increasing evidence that in mammals there are in fact transmeiotic epigenetic inheritance does exist. And I think the most interesting and rapidly rising area of this is the fact that there is clear evidence now, at least in rodents, of paternally mediated epigenetic inheritance, although the nature of the mark that's actually been transferred in the sperm from the father to the offspring is not clear. Is it a protamine? Is it a microRNA? It's probably not a methylation mark itself. And those who are interested in that subject, I'd refer you particularly to the work of Joe Nadeau, who I think has now convincingly shown that there's in fact a large component of paternal inheritance of epigenetic marks for several generations. There's also other mechanisms, and I won't dwell on them now. David Haig raised the issue of cellular inheritance. I'll just add to that the presence of cells and stem cells in breast milk that get incorporated into the thymus of the offspring. There's the issue of the microbiome, which many of you have talked about already. And then there's a whole raft of indirect mechanisms of inheritance, of which the most interesting, I think, is the recreation of the inducing niche in the next generation, which then leads to epigenetic changes, which leads to a change in the behavior of the phenotype of the animals that grows up, so it regenerates the niche in the next generation, which is effectively the kind of studies that Michael Meany in particular has done. And then we've got a particular interest in our laboratory, but I won't dwell on it today, of the interplay between these I will come back to it, of non-genomic inheritance systems and inheritance systems. Now, I think it was Randy in his presentation earlier in the meeting, or yesterday, who pointed out that we need to think across different timescales. And of course, what we're dealing with in all of evolutionary biology is how organisms respond to change, environmental change. And we can think about it in physiological terms as change in the short time frame, homeostasis, homeoresis, rheostasis, or we can think in terms of long time frames and natural selection. But in between, we have environmental change over perhaps the order of 1 or 2 or 3 generations, but is still yet reversible. And how does organisms cope with that? And that's, of course, where developmental plasticity and adaptive developmental plasticity has its key role. Now, I think when we're talking about how the developing organism responds to the environment, we need to think in terms of 2 categories. There are clearly insults to the developing fetus that totally disrupt the developmental program. The classic teratogens, thalidomide, maternal rubella, etc., which totally disrupt the organism's left to cope with the consequences as it come, what again Mary Jane West Eberhardt would call phenotypic accommodation. Or the organism has the capacity to respond to ecological cues by adjusting the developmental program, and that's what we would call developmental plasticity. And there are really fundamentally a limited number of mechanisms by which adaptive developmental plasticity can occur. It can involve epigenetic mechanisms, it can involve learning behaviours, and it can involve what is still probably likely underpinned by epigenetic mechanisms, changes in organ structure, size, and function. What we've done in our laboratory with Mark Hansen in Southampton is think more about adaptive developmental plasticity, and I won't go into a lot of detail now because I think many of you who are interested will be familiar with the argument now. With severe insults, an organism, a developing organism, and I'll focus here on the mammal for largely now, must respond immediately to have a chance of surviving, and in that case the fetus will either reduce its growth in response to a nutritional limitation, or it may even induce its own delivery in response to infection to have a chance of surviving in the postnatal world. There's also other classic examples, the altered metamorphosis of the spadefoot toad and so forth. But if the insult is less severe, or it's not actually an insult at all, it's merely a normative ecological cue, what the organism may do is change its trajectory of development in a way to try and promote its postnatal fitness in the environment it thinks it's going to live in. And this is perhaps the phenomenon with which we are most interested. Now I'm going to focus largely on obesity, because that's what my laboratories study, both in Singapore and in Auckland. But obesity at one level is relatively easy to understand. This is just the classic mismatch of living, particularly in the last 50 years and particularly in the last 20 years, in a world with an extreme level, an obesogenic environment with an extreme load of processed foods, which are likely well beyond our past evolutionary exposure to nutrition, which leaves us with a level of evolutionary mismatch. We also live much longer lives on average, although there's always been a proportion of people that have lived into their 6th and 7th decades, but many more people do, and so there's been little or no selection pressure against diseases that emerge later in life and do not affect reproductive success. And I would remind you that in fact, hypertension, diabetes, obesity do not affect reproductive success greatly until the disease is particularly severe. But we also need to remember that there's remarkable variation, just look in this room, between individuals and how obese they get in an obesogenic world. And explaining that population and individual sensitivity is quite a challenge. For example, all the GWAS work that's been done, and we're now up to several million people have been GWASed in relationship to BMI, and the latest revision by Spidiotis, which was in Nature a few days ago, has shown that despite all this GWAS work done on literally hundreds of thousands of people, to date people have been able to explain 2.5% of the variance in BMI based on fixed genomic variation. And even, they estimate, if you GWAS the whole world, we might explain no more than 20% of variation in BMI. Now, there are problems here because, of course, these GWAS approaches don't deal with the challenge of pleiotropy, and they are probably therefore underestimating effects. But nevertheless, the fact is there's a big gap between our understanding of the genetics of the heritability of obesity, which is not being explained by what we know at the present stage. So that led people to think about what are the other explanations for obesity. And clearly, if it's not solely in our genes and not solely in our environment, because there is a strong familial component, is there a developmental component to it? And of course, the developmental origins of health and disease paradigm is based on this large body of evidence that links both experimental, clinical, and epidemiological, that links the conditions of early life to later life outcomes. Now, I won't go into a lot of detail today, but for example, the work from the Gambia shows that depending on the season of birth, and there is a 15-year difference in life expectancy for people born in the hungry season, born in the winter season, but that difference in mortality is entirely induced after 20 years of life, 20 years of age. So the mortality curves do not diverge to 20 years after you're born. There are many things that have been found in the developmental oriented paradigm, but most of the focus has been on the metabolic complex of obesity, insulin resistance, and cardiovascular disease. And the kinds of data that's produced are shown in these in this work of David Barker's, where he showed that birth weight was related to the lifetime incidence of non-insulin dependent diabetes in 70-year-old British males. The story actually didn't start with David Barker. If one goes back far enough, you can see that Kermack was pointing out in 1934 that the conditions of that childhood health and infant health were major determinants of adult cardiovascular mortality. Then Dorner in Eastern Germany at the time pointed out that there was transgenerational effects of a number of influences, particularly diabetes, on the next offspring. And then there's the famous study from Ravelli and colleagues showing in 1976 that the offspring of the Dutch famine pregnancy, which was referred to the other day in a presentation, became obese as adults. Then there were a raft of studies, of which the one that gets forgotten, but is in fact the first definitive study, was a study in Minnesota which showed that children who were born to women who developed preeclampsia, called toxemia in those days, developed hypertension as an adult. And then later David Barker again showed that cardiovascular mortality in a British cohort was related to birth weight, and then their field started to explode. But despite all this data, the initial responses were entirely skeptical, if not antagonistic, to the concept that early life events might have a lifelong consequences. And despite the fact that between 1980 and now, or 2010, over 30 years, there was an enormous amount of data showing that there were relationships between early life events or early life experience and adult disease, acceptance and incorporation into policy and into medicine was very slow. And the reasons for that are listed on this slide, and I won't insult you by reading through the slide, but there were a number of reasons listed here which were the reason for this antagonism and resistance. And what I want to show you over the next few minutes, that each of those was effectively addressed using evolutionary medicine and evolutionary theory as the basis of addressing these objections. And I think the people doing the work didn't often know that they were using evolutionary thinking, but in fact the way they approached it actually solved and addressed the issue. First of all, a key observation which was actually there in the original data was that this was nothing to do with birth weight. And that was very important because one of the criticisms of the Barker argument was low birth weight only affects a small number of Western people, perhaps 5% of the population. But if it's all about low birth weight, it can't possibly have anything to do with this massive risk of non-communicable disease. But then that group showed directly that independent of birth weight, mother's energy intake in late pregnancy integrated with the weight of the child at 9 years, predicted carotid intimal medial thickness, which is the first, at 9 years of age, carotid IMT is the first objective measure of endothelial dysfunction, and which leads to hypertension. And this is present already at 9 years, and now I can tell you it's also present at 4 years of age. And a raft of data went on to show that, in fact, all these observations were largely independent of birth weight, not entirely, I'll come back to that in a moment. And that's why, sorry, I'm just trying to find the slide. On this slide, I circled this because I had pointed this out to David Barker in 1988, that in fact, even within the normal range of birth weights, there was actually something going on. So this was not a matter of extremes, but was in fact a matter of something that's happening within the normative range of fetal development. Well, we went on, and I'm not going to dwell you on the details now, but we did, starting in about 2009, we started looking on the basis, if this is a relationship, would we expect epigenetic mechanisms to be involved? And so we used one of the very early whole methylome array systems to try and identify genes, DNA in the umbilical cord, or sites in the DNA in the umbilical cord of babies, unselected babies, that showed great variance. Because we thought amongst those that showed great variance, we might then be able to back correlate that with the phenotypic change in the child. And to cut a long story short, we did this, we found a number of candidate sites in the methylome, of which might relate to childhood obesity. But when we went through replication and different cohorts and so forth, we focused particularly on RXRA alpha, which is a gene important to both fat metabolism and insulin sensitivity. And here you see that changes in the methylation in the umbilical cord DNA at birth strongly predicted children's fat mass at either 9 years of age or replicated in a separate cohort twice, and independent subsets at 6 years of age. And subsequently, we've done this again in an entirely different population, ethnic population, and are getting similar data. So we were excited by this because even though this came from a rather crude, unbiased approach, and we reached the RXRA alpha gene, this is a very strong effect, even though I wouldn't pretend on to estimate it precisely, compared to, for instance, what you see with fixed genetic variation. And we went on further, and we were able to show in one of those cohorts that the methylation change at birth was associated with mother's nutritional intake in early pregnancy. So what we had was a series of associations between maternal nutritional intake in early pregnancy. I'm not saying it's carbohydrate intake. That was what we measured. It could be some other aspect of the nutritional intake. The epigenetic change at birth in a realistically plausible gene leading to childhood obesity later on. Now I should say, and I'm not going to share the data, we subsequently, Alan Shepard in my laboratory has gone on to manipulate sheep in the period around conception with different genes involved and got exactly the same outcome. So it's actually been a test of the interventional manipulation, both of the epigenetic change at birth and the lamb's offspring outcome. But what we did in the human, well, what we've done, Alan did at the same time, was we took pre-adipocytes, we cultured them in various amounts of nutrition, nutrients, in this case looking at SAM-SAR ratios. And so we exposed the pre-adipocytes during differentiation to different concentrations of this one carbon metabolite in this one carbon cycle. The cells differentiated the same whether they had were exposed to SAR or not, but as mature adipocytes, we could see differences in glucose uptake, and excitingly, in the expression of RXRA alpha, the very gene we had shown in the umbilical cords, and changes in the very same region of the promoter, both in histones and methylation we had seen at birth. So while this is not a direct proof of a causal association, it is suggesting an interesting phenomenon that changes in methylation early in the differentiation of cells may not change the differentiation pathway, but may change the mature physiology of the cell later on. Now, this is not the only gene involved, I hasten to say. Here's another gene. There are a number of genes that crop up in this study. This is another one, and I only show it to point out that the methylation change is not always in the same direction. Here's a relationship between CDNK2A, which is an interesting gene I'll come to explain in a moment, involved in cell cycling, where the methylation change is inverse in relationship to the risk of obesity. But in this case, we actually were able to show both in the DNA, in the proteome, the methylation in this particular gene leads to different splice variants in genes associated with cell cycle function. And you can see that we could show both in the umbilical cord RNA, which I haven't got here, but in fat taken from adult, lean or obese individuals, change in the splice variants, in, the proteins and the genes and the expression associated with the splice variants according to this methylation. So again, these changes seem to be functional. So let me take all that and now turn to the evolutionary argument. The first person to try and put an evolutionary argument around this relationship between early life events and obesity was, of course, Nick Hales and David Barker, where they proposed the thrifty phenotype hypothesis, if you like, as a counterpoint to Jim Neal's thrifty genotype hypothesis. And they proposed that, based on the fact that they originally thought that this was a fetal growth retardation association, that the fetus became insulin resistant because it would have both survival advantage in utero and survival advantage postnatally. The problem with that model was this relied on the framing of programming, a term I don't particularly like but everybody uses, being related to growth retardation. And I've already shown you that clearly it's more than about growth retardation. It happens independent of growth retardation. And furthermore, those of us who, I'm no longer a neonatologist, but those of us who've practiced in the neonatal arena know that at birth, growth-retarded children are insulin hypersensitive, not insulin resistant, and do not become insulin resistant until about a year after birth, and we can talk about that later, why that might be the case. And more recently in Singapore, we've demonstrated using non-human primates that are growth retarded at birth. They also are insulin hypersensitive at birth and become insulin resistant about a year after birth. And so what started as an interesting evolutionary hypothesis needed revision. And of course, that's where I'll come to my contribution in a moment. But the other point that needed to be made is there's not just one pathway. There's clearly more than one pathway involved in the developmental pathways to disease. And one involves what I call the mismatch pathway, the developmental mismatch pathway, or the predictive adaptive response pathway, or the potentially adaptive pathway, about which I'll talk more in a moment. And the other involves a whole lot of evolutionary novel exposures. And the evolutionary novel exposures include maternal obesity, gestational diabetes, infant overfeeding, inappropriate weaning onto infant formula, and arguably a variety of environmental exposures. And indeed, if you look at the birth weight data in more detail, and this is more recent data from the American Nurses Survey, you can see there's a hint of this U-shaped curve, that there's something about fetuses that perhaps have a more deprived intrauterine life, and there's something about fetuses that have a more, if you like, nutritiously luxurious intrauterine life. And this wasn't seen in the early data because, of course, gestational diabetes and maternal obesity were not the issues they are now 70 years ago when those initial cohorts were found. So I'm going to focus largely on the first of the pathways, the predictive adaptive response pathway. And this is what Mark Hansen and I presented first in Science and then subsequently later, and we've refined it over time, the concept that the fetus who's subjected to normative ecological exposures, which will be across the normative range of nutrition, stress, and maternal undernutrition, which you regard as an ecological stress, I think, in evolutionary terms, will predict their postnatal life and change their nutritional and metabolic physiology accordingly. And again, I won't dwell on this at length now because I think those who are interested will have read about it. But what we've argued is that this is a developmental mismatch. Namely, the fetus predicts one world, a world of undernutrition, and ends up, for a variety of reasons, and ends up in a world with a lot more nutrition. And so we have two things in play, a developmental mismatch across the life course and an evolutionary mismatch over evolutionary time, and which has been exposed in the last 30 years, that compound to lead to the offspring becoming obese. But this is all hypothetical. Can we actually test the hypothesis? Well, to test the hypothesis first, we use rats. And it's relatively easy in rats to do this. All you have to do to produce developmental mismatch is undernourish the mothers during pregnancy and put the babies after they're born and been weaned on a high-fat diet. And lo and behold, you get the couch potato rat, which has all the features of the metabolic syndrome, except using a TV remote control. But interestingly enough, it has both central features, including hyperphagia and a fat preference in the diet, and it has the peripheral features, and we can show already at birth epigenetic changes in the livers of these animals. But what I want to do is just go through the experiment in a little bit more detail. These are the four groups of rats, and this is their adult retroperitoneal fat, their visceral fat. And what you can see here is animals are born to normally fed mothers and and on a normal diet after weaning. Animals born here to normal mothers and put on a high-fat diet after weaning, and yes, as adults, they get a little bit obese. Here are animals born to mothers that are undernourished in utero and on a normal diet after weaning, and they get about the same amount of obesity. But here, born to mothers that are undernourished in utero and put on a high-fat diet after birth, they get very obese. So the only differences between these two groups that are on a postnatal high-fat diet is what the mother's been fed in utero, and what's happened here is clearly the prenatal environment has sensitized the offspring to the obesogenic environment. And this has been repeated by a number of groups in different species, and the same thing has been seen. So what we did is we said, could we trick the animals that have been born to undernourished mothers into thinking they're born to a well-nourished mother than a thin mother? And we've done this in different ways, but the most famous bit of work we've done has been the leptin experiment, where we took these neonatal rats from undernourished mothers, where under normal conditions they'd go on to get fat. We injected them with leptin only between day 3 of life and day 12 of life, and then followed them till 170 days of age, and we completely stopped them getting obese. They look no different to control animals, and we blocked everything about these animals, including most of the expression and epigenetic changes in the offspring. So this, and we've done the same thing with neonatal growth hormone, suggesting that changing what the animal thinks it is when it's a baby changes the way its developmental trajectory goes. But could we do this in humans? Well, it's difficult to go around doing this in humans, but we thought about a population in Jamaica we were aware of, where we know that children who get kwashiorkor or marasmus, after they've been nutritionally rehabilitated, have very different nutritional controls. So a child who has marasmus, 6 months after nutritional rehabilitation, is able to mobilize their nutrients far better under starvation conditions than as a child who's had kwashiorkor. And furthermore, we go that children who've had marasmus go on to become adults who are stunted and have relative obesity and insulin resistance if they're in the conditions where they can get fat. And we thought about this because these two syndromes are syndromes of infant severe malnutrition. Marasmics are those that just waste away, looking like just getting thinner and thinner and thinner. They can mobilize their nutrients, but in general they survive. Kwashiorkors die from a breakdown of a whole lot of their bodily functions, but when they die, they've still got a lot of fat stores they haven't mobilized. And nobody's been able to explain the difference between kwashiorkor and marasmus. So we wondered if these were two different morphs, if you like. Those that they already preset before they were exposed to severe malnutrition to respond differently. Because the marasmics cope with the malnutrition, the kwashiorkors can't. Well, in Jamaica, there's been a very good nutritional rehabilitation unit. So there are a large number of children who while they get marasmus and kwashiorkor, they get rehabilitated. So even though under wild type, if you like, under normal, kwashiorkor children have a much higher mortality, about 10 times higher, in Jamaica, they all get rehabilitated. And we found a record on 1,300 patients that had born in hospital who had subsequently developed kwashiorkor and marasmus. And much to our surprise, we found marasmic children are born 300 grams smaller, which is an enormous difference in birth weight than those who develop kwashiorkor. So what we argue is the marasmics predict a low nutritional environment. They can cope with the environment better, but because marasmics have a much better survival advantage, they survive malnutrition where kwashiorkor's not. Does this actually support the predictive adaptive response model genuinely because of the fitness advantage in being born small in this environment? Now, I want to go on further and just again use this argument to test a further evolutionary hypothesis. We argued from first principles that firstborn children would be more likely to become hypertensive and obese and insulin resistant because they're more constrained in their fetal growth than second-born children. And the reasons for it is the uterine arteries don't dilate as much in the first pregnancy as they do in subsequent pregnancies. And in evolutionary terms, think about it. The pelvic dimensions are not maximal till 4 years after puberty, after menarche, whereas under presumed past conditions, most girls would have been mated and pregnant within 2 years after menarche. And therefore it makes sense in evolutionary terms to constrain the growth of the firstborn child more than the secondborn child. And given that now there's been this enormous demographic change, with far more of the world's population being firstborn, because family size is much smaller, could this be relevant, and could we find epigenetic changes that would explain this? Well, based on this theory, Keith Godfrey in Southampton went on and studied 30-year-olds who were either firstborn or secondborn, and independent of mother's body mass in first pregnancy, firstborn children turn out to be fatter. Then Wayne Cutfield in Auckland went on and shown that firstborn children by the age of 7 years of age are more insulin resistant than are secondborn children. We then did a 450K analysis, that's an methylation array, and much to my disappointment, we couldn't find any difference in the epigenetic state at birth. And this led me into thinking, because I knew I was right and there was something wrong with the 450K. So this led me into thinking for the first time about genetic epigenetic interactions. And we have a cohort in Singapore, an unselected birth cohort, where we have 1,000 DNAs at birth, which we've both done 1 million SNPs, and we've done the Infinium 450K. And on these pregnancies, we have a lot of information about the developmental environment. And all we did, Joanna Holbrook did, was she marched down the epigenome looking for areas where there was a lot of variation in the epigenetic in the methylation in the cluster, and then back correlated that with the 700,000 SNPs. And just to show you very quickly, because I'm running out of time, on the genome, the 3 ethnicities, because they don't intermarry, and this is on the initial 237, but it's true for the whole 1,000 neonates. On PCR, there's no overlap in the genotypes between the 3 populations. They're entirely separated. When we look at the epigenotype, you can see that separation is lost. And then we went ahead and we modelled what was explaining the epigenetic variation involving the SNP that had the highest correlation with the methylation site using a classic G times E models and interactions. And what we could see is in no case could we explain the methylation variance at birth on an environmental measure alone. In 25% of the cases, we could explain it on a on the SNP alone, and in the other 75%, the model involved a G times E environment. And just in general, just around the genes, the SNPs that are involved, there was very few that involved the CPG site itself, 13 out of 1,500, but about half the others were in cis, and obviously had tended to have stronger effects, but half were in trans. And I think this really highlights the issue. There's pleiotropy galore going on, and what we have is some really interesting interactions between the genome and the epigenome. I don't have time to dwell on it now, but when we then, and this I should say is a very preliminary analysis because we've not looked at the interactions across all these terms yet, when we looked at what was driving these differences in these 1,500 epigenetic states at birth, yes, the 25% that were genotype, maternal smoking, maternal age was important, birth weight was important, gestational age was important. Nutrition affected a number of genes, and of course, this is just the number of CPGs. We're not getting into the genetic pathway analysis at this point. But lo and behold, parity came up. And in our initial, and this just shows you this kind of magnitude of effect that we were getting. Here is a gene involved in metabolic control, the methylation here versus birth weight here, and you can see negative relationships for one genotype, or one haplotype, positive relationships with another haplotype, and for the heterozygote, absolutely no relationship at all. And the more we've looked at everything we look at, if you take genotype into account, you see very different relationships between the epigenotype and the environmental influences you're interested in. Our part of the analysis suggested 191 genes are involved now in parity, which if we hadn't taken genotype into account, we want to get into. I'm almost finished, Alan. By 2011, in my judgment, all those issues that I had talked about earlier had been addressed. Most of them had required evolutionary thinking to address them. And I felt that there were together, these led to a profound change in the way people felt about the developmental origins of health and disease, such that in 2011, at the United Nations High-Level Summit on Non-Communicable Disease, this clause, which admittedly I had some hand in writing, was inserted into the declaration. Note with concern that maternal and child health is inextricably linked with non-communicable diseases and their risk factors as specific as blah, 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 blah. The point being, by that time, the world's public health community, based on the kind of data I'd shown you before, suddenly said, hey, this might be really important. It should not be ignored. And this led, the result of that UN declaration, at the beginning of 2014, the World Health Organization set up a special commission on ending childhood obesity, which I co-chair with Tanya Nishtar from Pakistan, and this report was just released last week. And what I want to show you is a figure from that report, roughly, which, if you look at it, is basically a developmental plasticity evolutionary argument, namely that the organism is more plastic in early life, that things happen which change the sensitivity to disease later in life. And if I overlay on that the text of that document, you can see exactly an evolutionary argument in play about adaptive developmental plasticity. Things happen in early life which induce a different trajectory of development, as reinforced by what is happening in early childhood, and then there are different responses to the obesogenic environment later in life. And I genuinely believe that if it had not been for evolutionary thought entering this debate 20 years ago, we would not be at this point where evolutionary arguments effectively about developmental plasticity are being used in global public health arguments. So I leave you with that thought. Is development more important than we think in the evolutionary paradigm? I'm not going to dare answer that in front of the people in this room, but you might think what I think. So I'd like to thank everybody, and thank you very much.